Hello, everyone, and welcome back to the RNA-seq overview lecture. This is lecture 1 of 2 of the GL4U RNAseq module set, where we cover RNA sequencing and data processing. My name is Amanda Saravia Butler, and I'm currently the NASA GeneLab Science Lead. And so during this part of the RNA sequencing overview lecture, we're going to cover the information in part 3. Before I go any further, I want to make sure that all of you guys listening have completed and listened to part 1 of this lecture, also done the hands-on activity of the Jupyter notebook for RNA-seq processing through step 2C, covering part 1 through our trim data QC. And then I have already listened to part 2 of this lecture, where we cover alignment in QC, strandedness and quantitation in QC, and have also completed the GL4U RNAseq processing Jupyter notebook, so step 3, all the way through the end of that notebook, which I believe is step 8C, generating our genes.results files from RSEM. If you have not done that, I want you to pause this video and go back and do those things, and then come back and join us for part 3 of this RNA-seq overview lecture. And during this part, we are going to be covering our count data normalization, differential gene expression analysis, and data visualization. And so let's go ahead and jump to where we left off at the end of part 2. And if you remember, at the end of part 2, we were left with having generated our raw count data using RSEM. And so now we have gene quantitation raw count data for each sample for each gene of our reference. And so that means that we can go ahead and jump into the data analysis portion of this RNA-seq pipeline. So just as a reminder of where we are, we started off checking the quality of our raw reads, and then we went ahead and trimmed our raw reads to remove any adapters and low quality sequence data. We then checked the QC of those trimmed data to make sure that we did just that. We then, in the part 2 of this lecture, and in the hands-on activity, talked about how to align our data. So again, talked about different types of aligners, splice-aware, splice-unaware aligners, and then dove into splice-aware aligners, such as STAR, which is what we use in the GeneLab pipeline. These aligners require to build an index from your reference files. So reference files coming from the organism in which your samples were derived. So our hands-on activity, we're using data derived from mice. And so our reference files were from mice. So the mouse genome sequence and annotations. And we built a STAR index with that, just organizing that sequence and annotation information to make the alignment step more efficient. We then talked about how to align those reads to your reference, and then took a look at the log files of that alignment information to see how well our reads mapped to our reference, percent uniquely mapped reads, percent multi-mapped reads, etc. And then before quantitation, we wanted to make sure that we knew the strandedness of our data. And so we wanted to know if our data were generated with a stranded kit or not. And if it was stranded, which orientation relative to the reference it was stranded. So that way we could use that information to know how to quantitate our data. And then we talked about different types of quantitation tools, weighted versus unweighted, and talked about how we use RSEM, which is a weighted tool, which basically looks at the uniquely mapped reads, counts all of those as one, and then builds a maximum likelihood estimation to determine the probability of the multi-mapped reads coming from each gene or isoform that it was mapped to, and then splitting up that read proportionally based on that likelihood. And then that gave us our weighted count data. And that was the table that we just looked at in the previous slide. We will then import that raw count data. We use R in GeneLab to process our data. And then what we're going to talk about is our next step, and that is normalizing those count data, which we're going to have to do prior to differential gene expression. So let's talk about normalizing count data. So now that we have quantitative gene expression data, are we ready to run differential gene expression analysis? Not quite yet. So although we've counted the number of reads aligned to each gene, we haven't accounted for differences in read depth, gene length, or RNA composition. Just think about why this is necessary. So remember when we looked at our sequence data and we looked at our read depth, or the number of reads per sample, we saw that it was different for each one of our samples, right? And so we need to account for those differences. And then we're also might want to account for differences in gene length, and then we're also going to want to account for differences in RNA composition. I'm going to talk a little bit about those a few things and why we need to do that. So looking first at read depth, or the number of sequences we have per sample, accounting for sequencing depth is going to be necessary for comparison of gene expression between samples. So in this example, each gene appears to have double the expression in sample A, right? Double the expression in sample A than sample B. However, this is just a consequence of sample A here having double the number of sequences than sample B. And so if we didn't account for the fact that these were sequenced at different depths, we might falsely assume that gene X, Y, and Z are doubly expressed in sample A compared to sample B, or expressed at twofold more than sample B. So we need to normalize for those differences in read depth if we want to be able to compare the gene expression of each one of those genes between these two samples. Next thing we might want to consider, again, depending on what question we're answering, is accounting for gene length. So accounting for gene length is going to be necessary if you're comparing expression between different genes within the same sample. So within sample A here, if you want to know what the difference of expression is between gene X and gene Y within sample A, if you look at here, you can see that gene X and gene Y have similar levels of expression, right? 1, 2, 3, 4, 5, 6, 7, 8 copies, 9 copies of each one. However, just because gene X is longer than gene Y, gene X is going to have many more reads mapped to it than gene Y, just because this is a longer gene. And so if, again, we want to compare the expression of two different genes within a same sample, we're going to have to account for those differences in gene length. Next, looking at RNA composition. So for RNA composition, we can have a few highly differentially expressed genes between samples, and differences in the number of genes expressed between samples or the presence of contamination can skew some for RNA composition is going to be recommended for accurate comparison of expression between samples. And it's going to be particularly important when we're performing differential gene expression analysis, right? Comparing the expression between two groups of samples. So in this example, if we used a normalization method such that we divide each sample by the total number of counts to normalize, the counts are going to be greatly skewed in sample A because of this DE gene being really highly expressed. So that's going to take up most of the counts for sample A, but it's not crazy highly expressed in sample B here. So if we divide both of these counts by the total number of reads, the total number of reads in sample A is going to be more than the total number of reads in sample B. And so it's going to skew these data such that the denominator that these counts for these other genes are being normalized by is going to be so much larger that the expression of these other genes are going to seem smaller because of this highly expressed, differentially expressed gene. And so of these three different factors that we talked about, which one of them are going to be important if we want to perform differential gene expression analysis? Keeping in mind the differential gene expression analysis, we are comparing the expression of genes in different samples. And so we're going to want to account for both read depth differences as well as this RNA composition difference, keeping that in mind when we're doing our normalization for our read depth. So there are several different methods to use to normalize your count data. They are useful for different things depending on what you want to do with your data. So I'm going to talk about a few of them, starting first here with counts per million, or CPM. And just in this column here, saying the normalization method, then in parentheses is just an example of a tool that uses that normalization method. Again, these lists of tools, not by any means exhaustive. There are a ton of different tools that do normalization and a ton of tools that use similar normalization methods. I'm just giving some examples. So starting here with counts per million, or CPM, Limboom being one of the tools that do this. This is where the total reads in each sample is divided by 1,000,000 to create a scaling factor. And then gene counts are then divided by that per million scaling factor. Because you're totaling the reads in each sample, you're not accounting for read composition, but you are going to be normalizing for read depth here using that scaling factor. So it accounts for differences in sequencing depth. The uses for this would be to compare gene counts between replicates of the same group. So as long as you're in the same group here, you should be okay, but it's not for within sample comparisons or for differential expression analysis. Another normalization tool is reads or fragments per kilobase million, so RPKM or FPKM. RSEM is one tool that'll actually generate this. If you remember, in addition to our raw counts, we also had some normalized counts. This was one type of normalization. And this essentially just takes those counts per million, or CPM values, and then divides it by the length of the gene. And this will, again, account for differences in sequence depth with using that CPM. And then because you're dividing by the gene length, you are also normalizing for your gene length. And so uses here would be to compare gene counts between genes within a sample. Again, since we're not accounting for RNA composition here, it would not be useful to do between sample comparisons or differential expression analysis. Another normalization method is transcripts per kilobase million, or TPM. RSEM, Salmon, and Callisto are all tools that will do this normalization method. And this works very similar to RPKM or FPKM here. The only thing that's different is the order of operation. So whereas here you're doing that per million scaling factoring first and then dividing by the length of the gene, in TPM, you're going to divide by the length of the gene first, all of your counts, and then you're going to sum all of those RPK values in each samples, divide them by 1,000,000 to create your scaling factor, and then each RPK value, which is your gene length normalized count value, is going to be divided by that per million scaling factor. And that's going to account for sequencing depth differences and gene length differences, but again, not RNA composition. So this is useful to compare gene counts within a sample or between samples of the same group. But again, you're not going to want to use it for differential expression analysis because you're not taking into account that RNA composition. Other examples is the median of ratios method. So DESeq2 is a tool that utilizes this normalization method. It's actually the one that we are going to be using, that we use in the GeneLab pipeline. And how this works is a size factor is calculated for each sample by dividing the median ratio of all gene counts by the geometric mean of each gene across all samples. The raw counts for each sample are then divided by the sample specific size factor for each gene. And so by doing this, since they're using a median as opposed to like an average or a total, that means that you will not skew your data based on RNA composition. So you're accounting for that in your normalization. So this is going to account for differences in both sequencing depth as well as RNA composition. So this can be used to compare gene counts between samples. It's not going to be used for within sample comparisons, right? We didn't account for gene length here. And so it can also be used for differential expression analysis, though, so comparing the expression of genes between different groups or different samples. And then another normalization here is the trimmed mean of M values, or TMM. EdgeR is a tool that does this normalization method. You can also do the GE-TMM, which is TMM with gene length correction. And so what this normalization method does is the total read count, or total reads per kilobase, if you're using the gene length corrected TMM, is then corrected by a normalization factor, which is calculated using a weighted mean of the log expression ratios between samples after trimming. And then that's going to be scaled to per million reads. So this normalization technique accounts for sequencing depth and RNA composition. And if you are implementing this gene length corrected TMM, then you're also accounting for gene length. So you can use this to compare gene counts between and within if you do the gene length correction samples, and also for differential expression analysis. In the next lecture that comes after this, we're going to dive in a lot more to that median of ratios methods and show you some example calculations so that you can understand that a little bit more, since that's the method that we use in the GeneLab pipeline. So the type of between sample normalization used actually greatly impacts your differential expression analysis. So how you normalize the data has a huge impact on your differentially expressed genes that you end up getting. So make sure you're using it depending on which you're using the appropriate one to answer the question that you want to answer. As I mentioned, we're going to discuss DE-seq2 normalization method in greater detail in the next RNA-seq lecture, which is going to be a statistics overview lecture. And so after we normalized our count data, again, making sure for differential gene expression that we count for both, or normalize for both read depth and RNA composition, then we're able to do differential gene expression, comparing the gene expression between different groups of samples. And so that's the very last step of this pipeline. So after we normalize that count data, we can then do differential gene expression analysis. We also in GeneLab use DE-seq2 for differential expression analysis. And so what we do here is we're going to attempt to determine the probability of each gene's expression being significantly different in one of our groups of interest by pairwise comparisons, or you could do multiple comparison testing. And so in the example that we're doing in the hands-on portion, we're comparing flight versus ground control, so doing a pairwise comparison here. With DE-seq2, you can also do a multiple comparisons testing. Similar to all the other steps in our pipeline, there are several tools available for differential gene expression analysis, and they usually differ in the type of distribution that's used to create the model to fit the count data to perform the statistical test, and which statistical test that they use to do their comparisons. And we'll talk more about that in the next lecture. So here is just a few different tools, again, not by any means an exhaustive list. And for each tool, what the distribution or model is that it assumes the data to be in, and then the statistical test that's used. So DE-seq2, the tool, the package that we're using, assumes that the data are negative binomial, and it utilizes the Wald test for pairwise comparisons. And if you want to do multiple comparison testings, it utilizes the likelihood ratio test. Limavoom is another tool for differential expression. This one uses a distribution model similar to a t-distribution with empirical Bayes approach, and the test that it performs is a moderated t-test. EdgeR and BASIC also both use that negative binomial distribution or assumption. The statistical test for EdgeR is the exact t-test. The statistical test used for BASIC is a posterior probability through Bayesian approach. And EBSeq is just another tool. This one also assumes negative binomial, but it also incorporates the beta empirical Bayes model into its model. And the statistical test that's used for this is the posterior probability through Bayesian approach. And so we're going to dive in a lot more, particularly to DESeq2, this negative binomial distribution, and what the Wald test is in that statistics overview lecture, which is coming next. So hang tight if you're unfamiliar with those concepts. And so after we perform our differential expression analysis, we're going to go ahead and generate some visualizations from our data. And data visualizations are going to be used to help us interpret our count data and our differential gene expression data. And so in the hands-on portion, we're going to generate the following plots. We're going to generate a principal component analysis to see how our samples cluster in this case, two-dimensional space. So how they cluster, if do samples from the same loop cluster together and away from samples from other groups, or do they not? We're going to talk a lot more about principal component analysis, what that is and how that's calculated in the next lecture. We're also going to be generating cluster heat maps again to try to identify patterns within samples from the same group, and also how those patterns differ from samples in other groups. We're also going to be looking at volcano plots. The statistics overview lecture is going to walk us through the volcano plot in detail and how to interpret this. This is showing us the where our gene expression falls in terms of our log2 fold change of our pairwise comparison in relation to our statistical significance or our adjusted p-value. And then we are also going to perform a gene set enrichment analysis. So seeing if different gene sets are enriched in our flight versus ground control comparison is what we're going to be doing in the hands-on. And then we're going to visualize that using a dot plot and also by generating a gene set enrichment map. If these are unfamiliar to you, the next lecture is going to cover this stuff in detail. So this brings us to the end of our first of 2 lectures for the GL4U RNAseq module set. The next thing that you guys are going to be doing is you're going to be listening to the GL4U statistics overview lecture. This is going to get into some of those statistical models that I touched on here that we're going to be using for differential gene expression and how to interpret some of those visualizations. So very important lecture to take a look at before we dive in and do the analysis of our hands-on data. That lecture is going to be given by my colleague, Dr. Lauren Sanders. And so at this point, you guys can go ahead and stop this lecture. And then Lauren's going to take it from here and walk you guys through the statistics that we're going to be using in our next hands-on portion. So go ahead and get started listening to that statistics overview lecture. Thanks, everyone.